Okay, good evening, everyone. And again, welcome to this evening's seminar series. This is the African Science Frontiers Initiatives Science Seminar for 2022. So you're welcome. This evening, we will be looking at the topic you see in front of you, the issue of dementia. And I've invited my able colleague and friend, Dr. Thomas Karikari, who will be speaking. And then we'll have our moderator, Dr. Kenya Domeneko. So please sit tight and we'll get going in a moment. My name is Bright Waro, and I'm from Nigeria, but currently based in Sweden. So I'm coming to you from here. As you join us, we have everyone muted because we are recording these sessions. If the time comes for you to speak, we will, of course, allow you to unmute your mic and then speak. Okay, sorry, just having challenges with my. Okay. Here we go. All right, so this is our usual story. We start with to remind us where we are as Africans and as a continent, Africa. That if you compare Africa to other regions, of course, people may argue, why do you compare? But even looking within ourselves, we know that we are severely disadvantaged in many ways. We come from health indicators, technological indicators, different social parameters. We are severely disadvantaged. But it's a continent that has full potentials to be great. We are blessed from many fronts, but our problems are innumerable. So, what is the challenge? Many things happen in societies across the world, but one thing is sure, that any society that does not have a strong foundation in science and research will have challenges. And this is because science and research are key to societal development. And this fact is not new or strange to Africa. Africa remains the driver of much development and civilizations, we know, from the yesteryears. But we are now at the back of the driver's seat. So it is very important to remind ourselves so that we don't lose focus by putting our effort in different things that eventually do not give us the expectations, we desire for development. This text you have in front of you is UNESCO's mantra about what science is all about, what research is all about. It is there to help us to have longer life and healthier life. It is there to help us get medicines to cure our diseases. It is there to help us to get water and clean water to drink, to quench our thirst. It is there to help us grow our foods. It is there to help us get energy for our consumption, and of course, to have fun. If we do research and do science and it does not provide these basic needs, then we need to reflect, because science and research must respond to societal needs and global challenges. That is the basic mantra. But in Africa, our science has been stunted for various reasons. We can argue a whole day, we can stay and argue what the problems are. But the fact is that there are different fronts we have really pushed and made progress. And of course, we can think of how much progress we have retained. Independence, in terms of taking over power, was successful. We have had political revolutions as well. I have occurred, moving from military to democracy in many nations. Also have social revolution. And currently, Africa has a revolution in information technology that is really being positive. But one thing, or one revolution has not occurred in Africa. And that is what I think is scientific revolution. A system where scientists in the continent take hold of their destiny in their hands to make a statement that we have a stake to play in the direction where Africa's development should go. That has not happened. And I believe the reason for this is that there is a misplaced priority among scientists of what constitutes their mandate, what they should be doing, where their priorities should be. Misplaced priority happening. But for any society that has taken a center stage in the world, scientific revolution is part of that system. Science is recognized. And that is evidence even in the current pandemic we have faced or facing out. The COVID is a demonstration that science continues to be the voice of every societal development. So in ASFI, we believe that until such a time where there is a scientific revolution, a radical change of our current path led by scientists to revolutionize our thinking of issues that pertain to the African society and developing systems in which we can drive those issues to a solution that meets the basic needs of the African people. Until that time, we'll continue to remain in the system without making much progress. So it is important that we look inward as Africans, for Africans to put hands together to build Africa. Many times we have focused our attention on getting support from outside. I'm not sure how much that has really helped us. So we can't remain babies. We need to stand on our foot and then drive our destiny. And African science must play a key part in this. This is not a new system we are proposing here. It has happened and is happening across different nations. Singapore, India, has moved from the dentist to where they are today. China is an example, South Korea, and so on and so forth. Greg Mills aptly put it that poverty is now optional. So are stagnation and progress. So it is in this sense that African Science Frontiers Initiative, we call ASFI, was birthed as a platform for science capacity building in Africa. Our overarching goal is to contribute to Africa's path to development through science. As simple as that. Because there could be no meaningful societal development without science and research. But for science to play that role, it must be a thriving science. And a thriving science can only come through with the right infrastructure in place, at the same time as scientists themselves having the right competencies, having the capacity, and of course, being up to date with the knowledge and skills that are needed to address key questions that affect the society. So ASFI is a child of necessity, and it has been born to support African institutions, African scientists to develop competencies for high quality scientific research that particularly solves Africa's problem. So our vision is to raise the next generation of African scientists who have the right competencies to drive Africa's developmental and transformational agenda through innovative scientific research. We want to instill excellence in African science through competence acquisition, capacity building, and career development. We want to contribute to African science that tackles Africa's developmental challenges. But we believe that only a competent science can change our current narratives. So what we do is that we organize seminars like the one we are having today, workshops like the one we had last Friday. We organize courses like the one we had throughout last week, running each day 6 hours for one whole week to train, to motivate African scientists to do better. We have mentoring program. Each of our courses end up in recruiting new mentees so that the skills they have learned, we can help them to fine-tune them for better productivity. We also have research areas in which we dig deep into some of the issues to uncover the current state of the art in topical issues, and then use those issues that we uncover to develop programs that we can tailor our programs to. We target senior and junior researchers and PhD candidates. We take from PhD candidate upward. We have professors, senior lecturers from across African countries who come to our courses. We do not take master students or bachelor students because we have really lots of people who are interested in these courses, and we can't take everybody. The idea is to raise certain manpower that we believe can go back to their stations and contextualize and give back to the system they belong to. And once we continue to do this, we believe that the excellence we desire will trickle down to the whole system. When we started last 2 years now, we had a 3-year plan. And the 3-year plan is to train African scientists in capacity free of charge. So we have been doing it free of charge. Everything we do free of charge. But this excludes when an institution wants us to provide some trainings for their staff, then we do not do that free of charge. All right, so this is what we have done since we started 2 years ago. Yeah, this is not up to date. The course we had last week is not included here, but you can see the number that applied to our courses, the numbers that we have taken into these courses, and number of countries that are represented. So far, cumulatively, we have covered 35 African countries that have participated, participants have come from to our programs. Last week, we had a course on reproducibility in scientific research, which was second time we are offering that course. We had about 255 applicants, and we trained 120 of those that came to the course, and it was quite enjoyable, really, for all of us. Last year, we had the 2021 science seminar series in which we covered various topics, inviting Africans from all walks of lives, home and abroad, to share their experiences with brothers and sisters. And last year, our theme was disrupting stagnation, empowering Africa's future. That seminar series we had last year was a 3-weekly seminar series, which was enriching for us. So we'll continue. And one item we added to the seminar series last year was My Career, My Story, which was a session we invited African scientists who desire to share their experiences, their career trajectory and growth with Africans to motivate them. So I think this was really also very much enriching. Everything we do, our courses are recorded. The seminar series we have here is also recorded, and we have them all on our YouTube channel. Eman, my colleague, will be putting the link there on the chat box so that you can check them out, subscribe to this, and they like it. Share it with colleagues. I think you benefit from what we have there. So this year we have plans. One is the science seminar series we are having now. We have made it monthly seminar series in which we have captioned it spotlighting researches that are advancing Africa's development. So in essence, we're inviting people from all fields of endeavor and allow them to share with us what they are doing in their research area that are contributing to Africa's development. So we'll also continue our capacity building courses. We target this year to train at least 600 African scientists, adding to we have now trained more than 600 already during the last 2 years. We want to add 600 more to that number. And we'll be looking at the courses we have been doing and some of those you have suggested that we bring on board. At least the ones in front of you would target to have them this year. This one has gone. Okay, so please, if you have not taken advantage of these courses, do so, because by the end of this year, the 3-year plan, free courses we have planned from the beginning will elapse. Yeah, on Friday, we started the first edition of Dr. Wanro's research tips and tricks. We looked at how to formulate an impactful research question last Friday. And it was great to see almost 100 of you joining from all across Africa. We'll continue the next edition of this on the 18th of March, which will be looking at this important question, qualitative versus quantitative research, which is better? We know many of us are still engaged in that argument comparison. So come on the 18th of March, we are going to, within the 1 hour we have, to dissect this so that you go home equipped with tools to really deal with this issue. Which one do I take? Which one do I pursue? All right, so that is the introductory remark I want to make. And I would like to say a few words about our moderator for today, Dr. Kenya Omenoko. She's originally from Nigeria, but based in Dallas, Texas, in the US, where she's taking up her career and doing great things for Mother Africa. It's very impressive what Dr. Omenoko is doing. She had her bachelor's degree from University of Dundee in Scotland, and then goes on to Warden University, where she had her MSc in nursing, as well as her doctorate degree in nursing. She has done her postdoctoral research and now is a specialist in psychiatric mental health nursing and family nurse practitioner based in Dallas, Texas. She is a founder of David Omenoko Foundation, which she described to be dedicated to fighting the scourge and devastation of cancer in the society by creating awareness and advocating for cancer patients. I am personally aware of what she is doing in this area in her home country in Nigeria. It's powerful, it's encouraging. She has lifted the spirit of many people by providing opportunity for those who could not support themselves to screen themselves, treat themselves. She's doing great things in this area, and it's impressive. So this evening, it's my privilege to have her on board, to lend her voice to what we are doing. We are all pursuing the same mission to support our continent in any way we can do. So, Dr. Omenoko, it's my privilege to welcome you this evening to moderate this session. The floor is yours.
Thank you, Professor Nwaru. Good evening, good afternoon, and good morning from all over the world to our viewers. Like he said, my name is Dr. Kino Omenoko. I'm here from Dallas, Texas. And this evening, our topic and focus is a very interesting and a very important topic, put in a question format. Does dementia exist in Africa? This important and often neglected topic is going to be to understand the processing of neurodegenerative proteins in the human brain and biofluids, and apply this knowledge to develop new diagnostic tools for clinical use. To this end, he has been successful in discovering and developing tools to measure specific forms of the pathological tau protein in the blood. The first blood test he developed is now commercialized and is awaiting FDA approval for clinical diagnostic and clinical trial use. Dr. Thomas has received several major awards for these discoveries, including the Astrup Prize in Clinical Chemistry from the Nordic Federation for Clinical Chemistry, and the Best Alzheimer's Publication Award from the Swedish Alzheimer's Foundation. He holds research fellowships from the Swedish Research Council and the Alzheimer's Association and the Bright Focus Foundation, and is a principal investigator of grants from several other sources. He has published over 80 PubMed indexed peer-reviewed articles and has an awarded patent. Dr. Thomas was awarded his PhD in 2018 from Warwick University in the United Kingdom, funded by a scholarship from the Biotechnology and Biological Sciences Research Council, BBSRC. At the end of his PhD, Dr. Thomas was a visiting scholar to Harvard Medical School with his fellowships from Royal Society of Chemistry and BBSRC. Previously, he had completed his bachelor's education in biochemistry in his native country, Ghana, with first-class honors, and was awarded the Dean's Award for Academic Excellence. This he followed with his master's degree in pharmaceutical biotechnology in Leicester, United Kingdom, funded by a Commonwealth Scholarship. This evening, I have the pleasure and honor to invite Dr. Thomas to share his presentation. And before he gets started, I would like for our viewers to please put your comments and your questions in the chat box, and we will attend to them accordingly. Dr. Thomas.
Thank you. Thank you very much, Dr. Omenuko. It's a huge pleasure to be here today and also to be able to share my research. So, and thank you for the invitation, Bright and Eman, who organized this. And it's a huge lot of effort that you put into this. So I really applaud you for that. And in all the ways that we all can contribute, please let us know. So, we're going to talk today about quite, as Kenya said, something very provocative. That does dementia exist in Africa? And then looking at discussions on a neglected disease. How I'm going to take it from is from quite a biological, I would say, perspective, to trying to look at it from a clinical, but then more biological, and then there's been quite a few papers looking at the epidemiological aspect. I'll try to put them into it as we discuss. But trying to, in a link to the introduction that Dr. Ngwari started with, trying to link it to how could we improve the existence of this and the identification and the diagnosis in the clinical settings in Africa. So my name is Thomas Karikari. I'm currently speaking to you from Sweden. And yeah, I'm an assistant professor here, and I hold a joint appointment in the US. So sometimes I shuttle between the US and Sweden. So let's start. So the keyword in the title was was dementia. So let's try to work out what really dementia is. And it's quite a loose term that's often being used, that dementia. And I'm going to try so that we can bring it down. So it's an umbrella term. So it's often a broad term that's used to describe that. Often like in old age, people lose their memory and then the ability to think and to remember and to process new information, and it involves a lot. So like the broad term of dementia, when you look at it into from a biological perspective, there are different forms of it. The most significant part is the Alzheimer's, which is up to 80% estimated of dementia cases tend to have Alzheimer's. And then they have others: Lewy body dementia, vascular dementia, frontotemporal dementia, Parkinson's disease, Huntington's, and several others. And also you tend to have others that are mixed. So people could have multiple forms of these diseases, and that becomes the mixed dementia forms. And so what are some of the signs? I'll try not to go so much into it, but then mostly the diagnostic procedures in the clinic often bases on the signs that people show. That mostly is about reduced ability to process new information, memory, and declining memory, declining other forms of capability. So it could be like some people forgetting day-to-day things, language processing, solving new problems, or even old problems can be difficult, and that can be very simple problems. And then could have like some confusion and some personality changes, and it could be mood disorders and difficulty to perform pretty simple tasks as well, and also communicating issues. of course, there's no sense for alarm. It's not like if you have any of these, you have dementia, because we all forget things from one day to the other, right? And there are other things that we also have issues with. So it does not mean that if you have one or it happens, it means you have dementia. Actually, there are other diseases that are not dementia, but also we call them the transient forms. You could have some of these changes, but they don't necessarily mean dementia. So you have to have like a clinician follow you for a long time to be able to tell. So how big is the problem when it comes to dementia throughout the world? There have been studies that have been mapping up from different continents, and there's been estimated that, so like 2015, for example, throughout the world, there was close to 47 million people that were estimated to have dementia, to be suffering directly from dementia. And that in 2030, that is estimated to practically double. And that in 2050 is going to also double again. And it's the same trend when you look at all the continents. But there's a problem. One of the problems is that when you look at the case of Africa, these estimates come from the Global Burden of Disease, where they publish every year. But in the case of Africa, a lot of the data that they use in providing these estimates are not, I would say, are not correct. And I've written a commentary about this before, where they say that in a lot of the countries, including in sub-Saharan Africa mostly, they didn't have any information about dementia estimates. So if you ask me, like, what is the prevalence of dementia in Ghana or in Nigeria, there are very small papers that are out there, so you can't really tell the national estimates. So in these estimates are estimated, not from what would say like hardcore data, but from people's estimation. And we need to do better to try and provide, better estimates of dementia, and then to understand the context of it across the continent. One aspect, like, oftentimes when you ask in Africa, and I'm very interested for us to also start to study that, people would say, what dementia is just normal aging, right? When people grow old, they just have memory problems, they don't remember things. It's just okay. It's just a normal thing when you start to slow down during your aging. But that's like about 2 in 3 people, even in some of the developed continents, still think that that's the case. Well, from a biological perspective, and also from, I would say, clinical perspective, not everyone gets to have dementia. Of course, normally you get slowing down of some of your mental faculties, but it's not necessarily that everyone gets to have dementia, or that's the case. And also, about 1 in 4, so 25% of people tend to think that nothing can be done to change dementia. Well, we think that may also not be true, because more recently there are kind of some new drugs that are beginning to show a lot more capacity to improve dementia symptoms and to change it. So these are some of the things that maybe it's not just a matter of saying like dementia is there and we can't do anything about it. We might be able to do something about it. So back to this. I know I started with this term when I said, like, I know dementia, the umbrella terms, but then the majority is Alzheimer's. So I'm going to try and talk a lot more today about Alzheimer's, and that because people try to look at dementia is Alzheimer's is dementia, but it's not all forms that are dementia that are Alzheimer's. So Alzheimer's is a type of dementia, and that is a cause of dementia, because Alzheimer's belongs to the big term of dementia, and are accounting for that much. And I want us to make that clear distinction as we continue. And that someone will ask, what's actually the cause of it? There are two, as we know, as of yet, there are two separate types, as we say. Some are sporadic. Actually, the vast majority, up to 90% of cases of Alzheimer's, and I think it's similar for all the other dementias, we call them sporadic. Sporadic meaning that we just don't really know the causes. Of course, it's like estimated to be like a gene and environment combining and having effects, but it's difficult to pinpoint. And you see some articles, people say, "Oh, it's about your diet, it's about exercise, it's about this." From a scientific point, it's very difficult to say is one thing contributing, but that is also the majority. And we call also this late onset, meaning it shows up later in disease or later in life. So often start from 70, 65 years that people start showing signs of sporadic onset Alzheimer's. If you look at the familial forms, which means the people who have genetic predisposition, now there are some genetic mutations that make you very vulnerable to develop dementia later in life. And these ones include some of these genes, APP, personal one. There's no need to really focus on this, but just to say, like, there are some genes that when you have some known mutations, they put you at a risk to develop Alzheimer's dementia. And these ones are quite early onset. And the reason why we call them early onset is that they can show up earlier in life. So mostly like around 40 years, you start to show signs, clinical signs, compared to someone who, if when they develop sporadic, it's later, about 65. So you're about 20 years earlier. For example, there are people who have Down syndrome. Most of the people who have Down syndrome have a huge predisposition to develop Alzheimer's, and it's quite likely that majority are going to have Alzheimer's in their life. So it's one thing that some would say we should get tested, but also there are ethical aspects of it that we can discuss. So how are these diseases diagnosed, or the dementia forms, how are they diagnosed? And they, as of now or until today, the surest way that you can tell that someone has Alzheimer's or someone has a specific dementia form is when you do a pathological diagnosis. And unfortunately, that's when the time the individual has passed. And that the pathologist will take the brain and then they can take slices in this forms of way. And then when they do those slices, they can take them to perform staining, they do other pathological investigations, and then they can tell all those dementia forms that I talked about, which of them were present in the individual, and then they can provide a diagnosis. And it's quite similar to this. So this is how it's done. So this is a staining of that brain tissue that's been done, and this is a stain for a protein species, actually a peptide called amyloid beta. And this is how it is stained. So you have an antibody to this amyloid beta species, and then you stain the tissue from the individual. And that should give, whether it's positive, then that's how you get that kind of staining. And then there's also another protein called tau. So I'll come back and talk about this, like, there's A positive, it means amyloid positivity that for this amyloid beta peptide. There's tau protein, where you get to see this kind of also tangles, that's what they call them, that when pathologists we see these staining patterns, it means that the individual also had accumulation. And what we think is that these proteins do accumulate over time during the years. So they don't just appear, but it's an aggregation of things that have been happening over a long time. And one of the interests is trying to see how can we bring forward to trying to identify them early and also be able to start treatment also early. And there's another thing that when these things happen, if someone has Alzheimer's, they also, or any forms of dementia, they also to have shrinking of the brain. So for example, if you have someone who is like a normal individual, like this is how your brain, when the slices will show. But then if someone has these diseases, they also show a lot of shrinkage in specific regions of the brain. So they just start to, what they call atrophy, just start to lose parts of your brain. And these are all possible, but when, especially these two, can only be done when the person has passed on. So the problem is, do we want to wait until someone has died before we try to say, okay, they had this disease or that disease? Well, that would be helpful, but unfortunately, not to the individual and not to the family, and also not to science. Because if we want to do something, then we have to be able to do it earlier. So at present, there are different ways of diagnosing during life. And one of these, and now I'm talking about the word biomarkers a lot, because biomarker was a biological marker that basically shows it is a marker of something, is an indication of something going wrong or something going right. In the context of what we'll be talking about today, we're going to talk about as an indication of something going wrong. So during life, there's imaging markers, biomarkers, and for this amyloid, for the tau, and also for the neurodegeneration. Now, the amyloid part and also the tau part, the way they do it is that they've developed small molecules to the amyloid and also the tau, and that these small molecules you get to people are given in a liquid form, so you drink it, and then they go past the blood-brain barrier, they go to your brain, And that if you have aggregate of this amyloid or this tau in there, they will go and bind to it. Now, these molecules are also fluorescent, so they've been given their radio labeled so that they give color. And then after that, you go and do an MRI. So you go through the MRI, and then the MRI will image your brain. So this is what happens. If the person has amyloid, then you get to see. So basically, like, if someone with a higher extent of amyloid and looking at a cross section of the brain, it shows huge intensity of that ligand for amyloid. So there's amyloid PET, what they call it positron emission tomography. Forget about the long word. Basically, what it means is that someone has amyloid positivity in their brain. And it's the same thing for tau, for a different molecule that does that. If someone doesn't have tau positivity, it's more like this. When they have a lot of it, then it starts to show. And then for the neurodegeneration, which means that you degenerate in your neurons, in your nerve cells, there are two ways. Either they also do a similar method like these two, but then using a different radioligand that looks at glucose metabolism, so that when you start developing dementia, the metabolism of glucose gets to reduce. Or you can do a simple MRI, structural MRI, that will start to show that, for example, you're losing a lot of part of your brain as well. One is, these are pretty cool and they're very colorful and they're very informative, but there are two problems. One is that they're extremely expensive, which is that, for example, they're available in the US and in many parts of Europe. However, to have a single scan for these costs about $5,000. And not everyone's going to be able to afford it. And also, they can only do a few people per day. So it takes time. So the option is trying to look at if there will be other ways to improve it. And here comes CSF biomarkers. So the CSF is the cerebrospinal fluid, which is basically your spinal cord. So the spinal cord has direct contact to the brain. And that one of my professors that I trained and that I still work with here was actually a pioneer in this field, where he showed that when these protein aggregates build up in the brain, some of them get to be released into your spine, or like your cerebrospinal fluid in your spinal cord. So basically, it's in direct contact with the brain. So what happens in the brain? Some go out. So these can provide window information into these ones. And that the equivalent of the amyloid in the brain at autopsy, amyloid by PET, is also CSF, so amyloid in the CSF. And there are two different peptides that have been looked at. And then for tau PET also, you can have CSF, phosphorylated forms of the tau protein. And then for neurodegeneration also, you can have neurofilament light, so there are other proteins that for that. So just to go over this again, there's been a recent criteria for Alzheimer's. So because there were problems with diagnosing the disease clinically, and there are a lot of confusion between how someone is diagnosed clinically and someone is diagnosed using biological markers. So the Alzheimer's Association in the US and then the National Institute of Health in the US put together an expert panel about a decade ago, and they came out with what they call the new criteria. So that is going to be Alzheimer's is only based on biological evidence of the disease, and this is extremely important because based on the clinical forms where you look at cognitive changes, where problem solving issues, functional problems, and memory changes happen, these ones can be quite fuzzy. Like someone can have the same problems, but then they have Alzheimer's, or they have Lewy body dementia, or they have Parkinson's. They could really confuse. So it's difficult to use those testing ways. But then if you look at it from a biological perspective, much easier. So they have this ATN criteria, which is amyloid, as I just talked about, tau, or neurodegeneration. And in all of these, it's either you use the PET, as I just talked about also, where you have increase in sense binding of these radioligands, or of amyloid beta or tau, or MRI, where you look at atrophy of the brain. And it's the same thing that we can also look at using CSF biomarkers in the spinal fluid, that you can show that changes do happen as people start developing this disease. So one thing was the importance of these biomarkers, that how useful do they provide. One thing that we have been able to show, and that's quite nice, is that it appears that these biological changes from all the markers I just talked about, whether using the cerebrospinal fluid markers or the imaging markers, they all start to change like, from the CSF A beta or the amyloid PET or the CSF tau or the imaging tau, they all start changing way before clinical signs begin to show, which means that if we were fortunate and in being able to identify these earlier, then maybe we can be able to try to stop them earlier than they become clinically evident. Because the clinical evidence is when people start showing concerning signs at home. So, for example, you're with your loved one, you get to see that they have memory problems, they having issues doing basic tasks. Then you get worried, and then you bring them to the hospital, and that's when they try to do cognitive testing, and then sometimes they look at it multiple times and try to suggest that they have Alzheimer's or other forms of dementia. But with these markers, two things: they can help us to improve early identification, and it can also help us to improve the true identification, because these have been validated to give us what actually the brain, what is happening in the brain. They've been validated against neuropathological diagnosis. So one problem is that, and I'll take you a little back, in as much as that we have these markers doing a really great job, there's still another big problem, that they don't work for everyone, not in the sense of biology, but in the sense of access. CSFR is really good, but then you need, a certified clinician who has a good experience to be able to take a spinal tap. And also, it's not all labs that have access to do it. Actually, there was a recent paper that showed that less than 50 centers in Europe and America actually do CSF testing. So it just tells you that if you come to Africa as of now, as of what I know, no one is doing CSF testing. Although it's even a little bit cheaper, but no one is doing it. And then if you talk about imaging, that's even worse. To look at PET, it's a lot more expensive. And actually, the instrumentation for PET, I understand there are only about 3 centers in Africa that do have this, and that if you're going to go for one shot and you have to be taking $5,000, is a lot of difficulty. So this is where I came in, and that's where we start to think about it. And it was more of a dream for the field for like a decade, that what could we do? What if we went to blood? What if blood could help us to identify? Could we see some of these changes in blood? But then that was the problem. So why would we be interested in blood? This is what I just talked about. That for PET, they are expensive, they're specialized, limited specialized centers, and also they begin to show later in the disease because they require clumping of these target protein molecules to be a lot. So I mean, like, if they just start and they won't pick it, that you need a lot of them forming together for these molecules to bind to them. And that is what they call off-target binding, which means that it could show positivity when in natural sense there is no positivity. And that's like, you have to do an MRI also to do that. And then for CSF, the cerebrospinal fluid testing, also you have to take a spinal tap, lie in this position, and someone has to take your spinal tap. So it's quite invasive, and quite people don't like it as well. But if we want to go to blood, then blood needs to not just be another marker, but it needs to be very good, that comparable to the certified ones. And that's where we came in. So one thing that I propose is that blood will improve access, because when I look back in my village, there is no access to these biomarkers. These are all fancy, and that's why I tell my colleagues here that the ability of these markers that we have now, they are fancy things. We don't have access. And the way to improve access is to be able to tell, like provide a better estimate of dementia and Alzheimer's and all different forms, is to have a way that almost everyone can identify. If we look at the global map, these PET and CSF, the access is very limited. In Europe, in the US, and in Australia, that's where we have this. As even in these places, where I estimate is about 20% of the global population exists, it's not like once you're there you have access. For example, like in the US, you have to have good access to your healthcare costs. Your insurance should be really good to have access to these things. And so what I estimate that, okay, but what about the rest of the world? Where is rare? Like we don't have access to these things. So if we continue, as we continue to develop these new things, what happens to us? We need to also improve it. And that's where blood comes in. That if over 80% of the global population do not have access to the current tools that are used for testing the disease, then we need to do something. Now, if we use blood, of course, everyone knows blood, and blood is taken in every hospital, right? So it means if we move to blood, then there's a likelihood that we can improve access and actually make it universal. And that's where I'm going to. However, there's a problem that there are issues with blood that, it's really far away from the brain. If you're looking at the brain, and if you're looking at blood, like, you have to go down to CSF, the spinal tap, and also go into the circulatory system. That's the issue. So that's one issue. It's really far. I know the farther away you get from the target source, which is where the whole business starts in the brain, that's where it is that it becomes more difficult to detect, because the distance moves away, and then your analyte, which is the interested protein or whatever that thing that you want to measure, the farther away you move from the brain, also the more difficult it becomes, because the amount will start to reduce as you go farther away from the brain. And also in blood, things get cleared out really quickly. There's a lot of turnover of blood as well. But it improves your accessibility, because blood is more accessible. Everyone can have access to blood, and that it makes it more easier to work with as well. Of course, from a biochemical point of view, it also makes things complicated, because things can get chopped off and rearranged in all different ways that make it difficult to use. However, we still wanted to plow on. So this is where I actually came in. That when I started a postdoc, and that things were not really working so well on the project that I had, and then I started to play around with some other things, and the results didn't make sense. But then I kept looking at it, and I kept repeating, and then I was getting the same results. So I was like, okay, there should be something here. So I looked at this is the tau protein, just from this is the whole length of the whole protein. But just don't think too much about it if you're not from a biochemical perspective or a biological. Just say this is a protein, this is the length of the protein. Now in the brain, you will see this whole protein there. There will be some chopping off and some rearranging, but you see everything there. Now, what I found is that when you come down to cerebrospinal fluid, which currently that there are tools to use, the protein start to be chopped off. So the part here, you get to loosen it in the cerebrospinal fluid. You still also don't see this part, because it's also involved in the tangles that build up in the brain. And this is like, from the brain to cerebrospinal fluid. Now, we're wondering what would be seen in blood, and this is why I found that actually, when it comes to blood, things actually chop off a lot more. Now, this is one thing important. The current tools that are used in the clinic for the cerebrospinal fluid assays, which are actually being used for diagnosis, they target these regions. So the region that, and also means that there's been a lot of people trying to find those in blood, and they were not working. And the reason they were not working is that they are not available in blood, at least not in high levels. So what I found is that if you're really looking out for things in blood, you have to look at the other side of the protein, and that's where you'll find in blood, and that's what I use. So that's where these are going to be highlighted in a review that I've written for Nature Reviews, and that is not still in revision, is actually in press, so it should come out in a month or so. So we use that new tool. So I use that to develop a new method for phosphorylated forms of the tau protein, which we know is up in the brain. It comes to the CSF and also comes into blood. And this is one of the very first studies that we did. And I will just walk you through this a little bit. In FTD, which is like frontotemporal dementia, remember the very first slide, that there's some forms of dementia that were not Alzheimer's, and that includes frontotemporal dementia. It shows that the levels of this protein are quite low. In people who are in their young age, actually these are just university students, like 20 to 25 years, the levels were also low. But then also people who are like older age, like elderly adults, but then they don't have this amyloid protein. They are negative for that. The levels are there just about. But then when people are also like about 65 years to 70 years, but then they have amyloid levels. But then they have amyloid levels in their brain, but they don't have any clinical signs. So they can do everything fine. There's no problem with them. It pushes up a little bit, not so much. It means that clinically, if you bring such individuals to the clinic, it will show that nothing is wrong with them. But at the biological level, there's something of an issue. If people go into the MCH, which is MCH stage, the mild cognitive impairment, which is the early changes that can be seen in the clinic of cognitive changes that could predict that people are moving towards dementia, it goes up also a little bit. And then for people who have actually moved into the full dementia phase of Alzheimer's, then it goes up a lot more. This was actually a big deal in the field because we thought this was not possible. So once we found this and we ran this, and we also show that it correlates with the tau proteins I just talked about, the PET imaging that you have to use radioligand. So the levels that you see in the blood actually correlates with the levels of these stainings that are of this binding that you see in the brain as well. And at the same time, it correlates to the level of the amyloid radioligand activity also in the brain. And at the same time, it predicts outcome of, sorry, go backwards. Yes, it predicts outcome of PET, and also that the changes do happen much earlier. So one thing that I was also really interested is that we've shown that these work quite now, and then we've gone ahead to validate in a lot of dozens of cohorts. One thing that I'm interested in is that how do we validate in people who have actually passed on, because that's the surest way that you can tell someone has a disease. So if you take the brain of someone who has passed on, and then they do the staining, and then we link it to this blood. And luckily, we had a cohort like that. So this is a cohort from London where a lot of individuals had donated blood up to 8 years before they passed on. And then they also signed to donate their brain for research. So we took those ones, and then we took the blood, measured the blood samples, and then we gave it to the clinicians, and they mapped it to the diagnosis that they had identified. And I remember when we finished this first, my colleague told me, like, it doesn't look good, but I said, no, that cannot be real. Here's the problem. So the red is those with AD pathology, like so actually with biological evidence of amyloid and tau, which is Alzheimer's in the brain. Non-AD, it means those with other forms of dementia that are not due to Alzheimer's. And then control means that people who have normal aging. Now, if you plot according to, this is 8 years, so blood measured 8 years before death, 4 years before death, and 2 years before death. If you plot according to the diagnosis that the clinician provided, it shows that the marker doesn't perform well. But if you plot according to the diagnosis, the true or the correct diagnosis given after death, like given at postmortem, it shows that the biomarkers perform really well, and that they can able to differentiate between those with evidence of Alzheimer's from those with dementia but not Alzheimer's dementia, and that those who passed on but did not have any dementia in their brain. Actually, we even went ahead to show that those individuals who were like overlapping here, they were given non-Alzheimer's dementia diagnosis at death, but they also had Alzheimer's in their brain. So their primary diagnosis was not Alzheimer's, but then they had secondary diagnosis of Alzheimer's. We've gone ahead to validate this in several other cohorts where we showed, for example, in some of these new tools that we've developed, when we look at the amyloid, as I talked about, the amyloid beta in the brain, there's another pathology cohort. It showed that there are increases in a huge number of individuals that we studied. Each point is one individual. And these new tools perform really well compared to some earlier tools that were developed that they don't show any difference. And this for amyloid, and also this was for tau tangles that we show that increases do happen compared to other tools that were developed earlier than this. And one good thing that I find is that the levels of these markers also predict longitudinal changes in cognition. So it means that if we were to measure these markers in someone's blood today, the levels would predict how their cognition, their memory problems are going to go. That, for example, if you look at these ones, those blue that the levels are lower, they tend to stay quite long for over a 5-year period. They don't go down with their cognition, their memory. But then for those in red, where the levels of these markers were higher, the same thing here, they tend to go down much faster. So it means that they're likely to deteriorate when it comes to the cognition much quicker. And we've also validated that because from what we developed, there were a lot of interest from pharmaceutical companies and other, so a lot of people jumped on it. But then one good thing is that we did a head-to-head comparison recently, and basically what we showed is that these tools that we developed compared to those that were developed by companies and pharmaceutical companies, they basically perform the same way. to identify Alzheimer's, to identify amyloid and tau. So one question is this: what's the importance? If we've identified these things, what could they do? One importance is that they will help to provide a confirmatory diagnosis of Alzheimer's. I've talked about earlier that you could have some individuals who come to the clinic and then you do cognitive testing. It shows that they are cognitively impaired, and you think based on some other aspect they're likely to have Alzheimer's. Well, now you could have a blood test that you can run as well, and it will provide a good estimate. So, for example, if someone has cognitive impairment and they have Alzheimer's cognitive impairment, then they're likely to have their levels of these phosphorylated markers go up compared to those who are cognitively unimpaired and don't have any evidence of amyloid or so in their brain. So it can help you to classify. And also, of course, there'll be other groups, like someone showing early signs of cognitive impairment, then they will sit here in the middle. So like it shows a continuum where things go up. At the same time, you can also do differential diagnosis, where you can show those who Alzheimer's diagnosis, these markers go up, but then those without Alzheimer's diagnosis, Alzheimer's dementia, so dementia due to other causes, then these markers do not go up. So you can split between them. At the same time, we can do a population screening, because there are a lot of massive epidemiological studies that have been done in the past, but unfortunately, most of them, almost none of them, had the capacity to include blood testing, because there were no blood tests in those times. But some do have blood collection, so we've actually started reaching out to some of them, and then we are collaborating to see if we can run in these huge studies, thousands of studies of individuals, and then we can map to show those with real evidence of Alzheimer's dementia. And one very important aspect is that in addition to clinical, the population studies, we can do clinical trials where we can improve diversity, because currently clinical trials for new disease and new therapeutic treatment, basically for new cures, have been tested mostly in white people, even in Europe and in the US. Now, because one of the issues is that a lot of minority people have concerns, of course, validated concerns from historical context, when there are conditions that they have to spend a lot of time. They also have to donate, go through some invasive procedures. Then there's a lot of trust issues. But blood is more acceptable, so hopefully we can start to expand. Now, this is what we think that these new tools could be used in the clinic. And these include that we're not saying that these should change how clinical diagnosis is done. What we're saying is that these ones should enrich how diagnosis is done. So normally, if someone is suspected of having Alzheimer's dementia or any forms of dementia or cognitive impairment, then you'd bring them to the clinic. And then you have a cognitive complaints. Then there will be an interview, about how long the problem has been, and also, their features, whether their age, their sex, if they fit the criteria. If they have family history of dementia, then it would be likely that maybe they'll have genetic mutations. Those mutation screening can be done. And then if there are other conditions, medical conditions that make them vulnerable to these ones, and then they can run other lab tests to rule out other causes. But then if for all of these, because an important thing here is that there are other conditions also that can present like this. But for example, if people have depression, some could have memory issues, but they don't take long. They tend to clear quickly compared to those with Alzheimer's. If the suspicion still persists, then you can run to improve what we call improving the diagnostic workup for the doctor. Then, in addition to doing a neurological exam, where they can do the MRI and all those ones that I talked about, and then they can do neuropsychological screening to provide where they are cognitively. You can also add blood tests, and one of the important blood tests that we want to look at is blood phosphorylated, because at present is the most specific for Alzheimer's dementia. And that's so in the new test that we developed, if you test for those ones, then based on the results, where the results sit, whether it's super high, whether it's gray zone, medium, or low, then you can tell where the individual should go. So for example, if the levels are high or close to being high, then it's likely a cause for concern. So you can refer that individual to a specialist, whether a geriatrician, whether a neurologist, whether, whoever is in care of that dementia care at that hospital is where they can go. And at that place, they can whether confirm CSF or PET testing, and then also looking at whether that individual can be recommended to go on clinical trials. And then you can also try to work it out to provide differential diagnosis, whether the cause of diseases are Alzheimer's or non-Alzheimer forms. At the same time, when the levels are low, then you could try to triage, what we call with neurofilament light, which is another blood protein, a blood marker, which is increasing all forms of dementia. So then you can also work that out as well. If neither of these shows concerns, then you could keep that person being monitored at the primary care, at your GP, at the healthcare level. Yeah, I think this also goes over the same thing. Another thing that we think these new tools provide is the potential for clinical trials. And this is one thing that we modeled, that we showed from one of the big studies that we did, that for those who have increased levels of these phosphorylated type protein we talk about, a lot of them also tend to have a myeloid PET positivity based on the imaging. So at present, for clinical trials, they require people to have increased amyloid PET positivity. And that is a big barrier for a lot of minority people. And also, it's a lot of cost as well. And again, it's also a lot of inconvenience to people who volunteer their time to be part of these studies. So on a normal day, if you're starting a study, and then if you're interested in having 1,000 people, so you have a new drug that you've developed as a pharmaceutical company, you want to test it in a clinical trial, then if you want 1,000 people to be part of it, then based on the results we have seen, then out of these 1,000, we estimate that about 20% will also going to have increased levels of phosphorylated tau. The reason being that these new markers change earlier. These blood markers surprisingly change earlier than the brain markers, actually the imaging or the markers as well. As I said, because the imaging ones take longer, they need to clump up together for a long time before you can see them. So we said, like, if for that 20%, instead of screening to get 1,000 people, and then to get 1,000 people, 20% of whom are myeloid positive, then have to screen minimum 5,000 people. And if you're going to screen, and we even went for like a very simple approach, that if you're screening for each individual, you're going to cost you $3,000. Some go higher than $5,000. If you're going to screen 5,000 people at a cost of $3,000, you're going to have like a huge cost of $15 million. But even aside from the cost, the inconvenience, and also the difficulty in having a population that's reflective of what is it in the real world. This is what we propose, that if you start with these new blood tests that we have come up with, then what we showed is that about 40% of those who are positive for A-beta PET also be positive for phospho-tau, also be positive for amyloid PET. And that you basically would just take that same 5,000 people, instead of screening them with imaging that they have to go to the clinic, then you can just screen them with a blood test. And the blood test is likely to show that 40% of that 5,000 people will have increased phosphorylated tau levels in the blood. And that's going to be super cheap. We estimate it like just going to cost $50 to do per individual. So you're just going to use a huge fraction, just a tiny fraction, $250,000. And that you then will take these people, like about 2,000 people, and then screen those ones with a myeloid PET. And that would improve not just the financials, but also the ease, the convenience for the people that you recruit. And hopefully these will be more attractive to minority populations, like people from African descent, people from African backgrounds, to want to participate. In as much as we've studied a lot of populations and also a lot of cohorts, we've studied like tens of thousands of blood samples from real individuals for these tests. We still feel that a lot needs to be done. And one of those things that need to be done is that most of the people we've studied come from American or European background, so people of European descent. Even in these continents where we have a lot of African people, a lot of people from Asia and elsewhere, they still do not reflect a lot in the cohorts that are recruited in the clinics. And one thing that I've been trying to push for is that before we can move this to the clinic, and these tools are being picked up really fast, They're already being used in the clinic in some cases. Instead of going so fast, we should also validate that in a huge population. And this is one thing that should be done, that we should try and work out when someone has an increased levels of these blood phosphorylated tau proteins, what does it mean? And before we can say what it means, we have to look at a lot of different populations, lots of different people. And that we should try to define for clinicians that when they have these increased levels of markers, what does it mean? Does it mean that people have amyloid? Does it mean they have Alzheimer's? What do they mean? And is it likely that these ones are also going to influence the diagnosis that the clinician gives as well? And that we should try and validate a lot of them with the established tools that are already being used in the clinic. And one of the ways to look at is that one thing that we know is that these markers also have other functions. So they are not just related to disease. They actually have normal functions in the brain and elsewhere. For example, the tau protein has functions for development. So when you have in the embryos have tau protein high levels, early in life they also high levels because they have to improve some neurodevelopmental functions. And after that, they go down. They only start to come up later, and some only come up later in old age when and if the individual has Alzheimer's. So we have to work it out. For example, do we need to look at age effects? For example, biological effect is not due to Alzheimer's disease. We have to look at them. Is there a sex effect? Is there a genetic or epigenetic effect? Ethnicity, other medical conditions as well. Cardiovascular, we have to look at all those ones. We've looked at ApoE, which is a big genetic risk for Alzheimer's, and kind of shows a tiny increase. Predictive capacity. And then for lifestyle as well, if someone is having other medication use, does it lead to a false positive for these ones? And also if the other factors, because if you're looking at blood, the problem with the blood is a huge traffic. There's a lot that's coming in, a lot that's going out. So are there anything that can affect these ones as well? And also the functions of the liver, functions of the kidney could be, or could be diabetes as well, could they have effects? Now, one of the very early stages, early things that we have looked at is trying to look at people of African descent. What we saw, so we're trying to look at multiple. It's difficult to get cohorts, to get studies of African populations that have access to these samples and are well done, and that we want to look at them. But we tried to do it. So the very first study that we did was to say that in the US, where they took people from African American backgrounds and people who are white people from Caucasian backgrounds, what we saw was that if you the people for the same sex, the same age, the same cognitive abilities, and the same genotype of this ApoE gene, that in white people is known to be a good predictor, a risk factor for Alzheimer's. And I collaborated with this neurologist who is in Washington University in St. Louis, Missouri. And what we did was to take this one and measure these same levels for the same. So they had CSF and they also had blood. And actually the results kind of were a bit staggering, that we saw that it saw like a bit of differences in the actual levels for the African Americans and then the non-Hispanic whites as well, both in the CSF markers, in amyloid PET markers for the brain, and also to an extent in the blood markers. And what we saw is that if you want to use these blood markers to predict positivity of brain amyloid, then the performance was lower in the African American population. The reason being that these cutoffs that have been developed were all done in white people. So it makes it more difficult. It means that if we've seen any tiny difference, then it's likely that there's going to be a lot of bias, a lot of disproportionate bias against people of of Black descent. However, this is not conclusive yet, because we saw in the African American population in this study, that a lot of them also had diabetes and then hypertension as well. We don't know if those have anything to do. And I'm really ambitiously trying to propose a study where we study thousands of individuals from all kinds of backgrounds within the US, in Europe, and also the equivalent populations back home. So for example, if we take an African American cohort, we also would take people, Black Africans in Africa and people of Black African descent in Caribbean, so that we can try and work out the same we do for people from Chinese backgrounds, people from all other backgrounds, and then we try to really provide a comprehensive approach to show how all these markers work. One other thing that we've tried to do, we still say blood is easy, right? But there's still a problem. I mean, recently there was a pandemic, but we're not even totally out of it. And in those times, you couldn't get people come to the clinic for you to take blood. And also, when you take blood, you have to store it in cold conditions. Well, electricity is an issue in some places, including ours. So this is one thing we have to look at. If some also are going to remote sites like field work, and people living in the hinterlands to take samples, how do you have to carry them with you to a center, and that will take hours. How do you work that out? In places where shipping is not reliable. So we tried to also model this recently. And one thing that we tried to do was that there's this commercial thing was called Noviplex cards, that when you take blood, you just take like from a dip and then you dip it on that. The separation of the blood into different parts. When you take the whole blood, it will separate into red blood cells, into the Buffy coat, and also you have the plasma or the other part at the top. This does the separation within. So you basically just take the blood, you dip it on it, and then you store. You don't have to do anything. And then the normal dry blood. So basically, you have a filter paper. You take blood, you just spot on it, it dries, and then you just will store it anywhere you like. And then after some time, like which will be in the lab, then you put it in the buffer, it redissolves, and you can have it. Another way is like if you delay when you take the blood, when you have to spin it so that it separates into the different forms, if you delay it for 2 days, what happens? We tried one of the new markers, neurofilament light, which is a general marker for all dementia forms. And what it showed is that in all these three conditions, if you compare to the normal standard of truth, where someone comes to the clinic, you take blood, you process quickly, you store it in the freezer. If you compare to those, although the absolute levels do change, but then there's a high level of agreement between samples that are processed this way compared to samples that are processed by the normal standard of truth. So, linking back to what Dr. Murray said, there is no escaping for us. There is no excuse. We have to be able to do these things as well, regardless of the condition of the settings that we have. So I'm going to finish off with what we are proposing to do in Africa. And as called the Africa Dementia Study, we're actually going to, there's another name I'm thinking of for it. As it stands now, there isn't a lot known about these diseases, and especially from a biological point of view, we don't know much. So what I'm proposing to do, and I've reached out to a lot of several experts in the field in Africa, and I'm also open to anyone who is really interested to collaborate in this, to try and look at what are the pathologies. So that, as I said, the best way or the surest way to tell what forms of dementia or any other brain disease someone has is to look at their brain after they've died. So in terms of, if you look at some brain banks, and I've reached out, and I know there's some pretty good ones in Nigeria and also in Ghana that we can collaborate with, that if we can do a postmortem analysis to look at what are the forms of these protein pathologies in the brain, and that at least will be a first step in providing us with epidemiological scale, that to say, okay, this is what we have. All right, if you take this country, these are the forms of dementia that are common, and that. And then you can also looking at profiling based on the genetics and based on the proteomics of the brain of what is in there. For example, if someone asks you what are the number of people with like a certain mutation for disease of any type of dementia, we don't know. And that's one thing we can do. And then of course we can bring in the blood markers to try and improve how we can work out the brain diseases, and also improve their clinical diagnosis, and also how we can use them in population studies as well, and in clinical trials. Last but not the least, we're looking at improving the epidemiology of disease of dementia forms in Africa, because as it stands, we don't know what it is. Like, what forms of if we take people over 60 years, what is the percentage of people with Alzheimer's or any forms of dementia in Ghana, in Nigeria, in Egypt? In the estimate, people put estimates, but they're actually just very wild guesses. And can we improve these ones as well? So yeah, I'm going to end here and say, okay, we've looked at new blood markers. They're really, really promising to improve the identification of biomarkers in the field in terms of like disease for dementia and other forms, and that these improvements will help provide a better characterization of the disease, validated against markers that actually come from the brain. And that what we're trying to do, hopefully, in Africa, will really, and it's one of the things I'm extremely close to my heart, that if we can provide a better understanding of these forms of dementia in Africa, and then that will provide a good way for us to diagnose better, and then try to also solve it earlier. And these are things that we should all join together. So I'll end here, and there's a bunch of people who have all been contributing to these works here in Sweden, also in the US, and my collaborators who are spread around the world. And yeah, I'm also actively recruiting graduate students and postdocs. So if anyone is interested, please reach out and we can have a chat on that. So thank you very much. And these are my email addresses as well. Thank you.
Dr. Thomas.
Dr. Menako, you're muted. Can you unmute yourself, please?
Okay, Dr. Thomas, thank you so much for this wonderful presentation this evening. We appreciate all your work. It is amazing, what you have done so far. And I'm hoping that the global world would listen and help you so that you can get to your destination. Dementia is a major problem all over, and like you've just told us today, there is still a lot that is not known about it. And the only way that we can find out is through research. And Professor Nwaro has this platform for sharing this information so that people can get the information they need and they can help with the resources that is required. And without people participating in research, then there's going to be, the problem is going to continue. Like you said, we're not going to be able to identify these issues, find the solution to them so that we can help ourselves back in Africa and the general population. So here we have it. We need to create more awareness about dementia in Africa. It is real. It is not your neighbor. It's not somebody, has done something to your family. It is real. So we all need to participate in our own way, whether we're participants in the study or whether we're the researchers. And we also need to collaborate. We need to also get the political commitment from the government and public health, so that we can prioritize these issues, bring it to the limelight so that we can continue to find solution to such problems. We are now going to take some of your questions and share some of your comments. So the first question, this is for Dr. Thomas, but cerebrospinal fluid is invasive, right? Why not plasma levels of these biomarkers? Is this option available in Africa, which you've already touched? So I'll let you say more on it.
Yeah, I guess this was an earlier question, maybe when I was
right.
touching the CSF part.
The second question is, how many transcripts from tau protein in humans? Is it only expressed in the brain?
Yeah. Someone taking me to the biochemistry and the biology. So there are different transcripts of the tau proteins, and it's quite an interesting protein that I've looked at. So in the adult, and I'm trying to be, so in the adult human brain, you have 6 transcripts. But then in children, so as I said, it has functions in development. So it has its own normal functions, but when things go wrong, that's when it starts to do bad things. In children, it has 1 transcript in the very early stages because they have their own function. But then when you grow older, then more transcripts are added. In the adult, by the time you're adult, you have 6 transcripts in the brain. However, there's another transcript in the periphery. So in peripheral sources, like in the kidney, in the heart, in the liver, you also have tau protein in there. But these ones have different, an extra exon that's transcribed in there, so that you can differentiate between tau coming from the brain and tau not coming from the brain when you want to measure them in blood, which is very, very helpful in this scenario.
Wow. Thank you. The third question is the blood kit readily available?
Yes, so at present, there are kits that are readily available from some of the pharma and biotech companies. However, you would need the platform to run it. So, for example, it's not like an ELISA where you could just read with any plate reader. The current one, the very first one, is Phosphorylated 101, is available on the Simoa's single molecular array platform, which is from a company in the US. Is really widely available in a lot of the big labs across Europe and the US. The platform by itself, so the instrument itself, cost $200,000, I think, to buy the instrument, and then you can buy the kits, and then you can run on them, and it provides you the information.
Okay. Thank you for that answer. Let's take another question. Apart from biological markers, are there cognitive tests to predict those that may be at risk of developing dementia in the future?
Yes, there are, and this is a topic that I've had discussions with just in the past weeks with some of the very revered people when it comes to dementia in Africa, including Professor Adesola Oguni at Ibadan, who is actually a godfather when it comes to dementia studies in Africa. Now, one of the issues is that, yes, there are cognitive tests, but as usual, these tests were developed in Africa, in Europe and the US. Now, these cognitive tests are based on what I call familiarity, because in there you have things like you're asked to do a task or you're asked to identify an animal. If you're not used to that animal, you can't pick it out, even in a normal day. So it's not like then if you get it wrong, it's marked as being impaired, or it contributes to being marked as being impaired, but in reality, you may not be. So they worked out in trying to adapt some of these tests to African settings, and there are some that have been validated in several African countries. However, one thing that I'm also interested in is that still it's difficult to remove education from this. So, like, you have some of these tests that do not require anyone to have a reasonable level of education to perform, and some of those tests have been done. So, for the study that I was talking about, we're discussing trying to combine some of the tests that have been done and validated in Africa, so that we can use in this proposed setting.
Right. Thank you for a wonderful presentation of that answer. This question, the next question, we're building a cohort study on dementia. This is where I comment. We're building a cohort study on dementia, and our plan is to collect some samples to assess the levels of A beta 1 to 42, A beta 1 to 40, total tau, and APOE. Do you have any advice to ensure accuracy in our measurements?
Yeah, and then depends on where you plan, and this is extremely exciting. Depends on where you plan to do them. I'm sure, like, the people you plan to collaborate with will have good expertise in doing that. But also, this may come quite straight. But if you're interested to collaborate with me, I'm also very fine to do this, and then we can bear some of the costs and then do this together.
Wonderful. I am interested in beta amyloid inhibitors, and I am now developing a protocol using bioinformatics approach at the first level. I guess it's more than a comment than a question.
Yeah, so I guess
any input?
Mhm. Yeah, so amyloid inhibitors are pretty exciting, as also, and as just to say that recently there was an amyloid targeting drug that has been approved by the US FDA for Alzheimer's. There's a lot of controversy around that, but there are other ones that we know of that may also be approved pretty soon that also target amyloid. So amyloid inhibition is kind of proven to be a useful approach, and it sounds like is a good way to keep going. So as you get bioinformatic data, it might be worth also validating with um, experimental data as well.
Okay. This one, thanks, Prof, for your excellent presentation. Very good initiative regarding the Africa dementia study. I was just wondering how you plan to break the cultural barrier regarding the conduct of such pathological diagnosis on corpses. How easy will it be, or has it been, to convince relatives to consent to using their loved ones' organs?
Yes, so yeah, this is a very important one. One thing that we are already doing is, as I say, like, collaborating with people who do this, like who have done this and have experience of doing this on regular basis within the target countries, and have the know-how of how to speak to the families and how to let them get them to consent to do this. So of course there's a lot of issues around that, but hopefully we will get enough people who would be willing to contribute. And of course, maybe we can also try not to have it as like, take the entire brain, because we actually don't even need it. That one of our approaches is try to do biopsies. So for example, you can, so like it's not so evident that part of the individual's brain has been taken away. Of course, under consent.
All right. Thank you.
That is the last question.
Yes, that's the last one that I have.
Yeah. Thanks a lot. Thanks a lot, Dr. Karikari and Dr. Emanoko for this evening. I think this is an eye-opener for many of us. There has been some interest, as you can see from the chat. I've put your profile on the chat box. So for those who are interested, there are a couple of people who have some projects Dr. Akari has said are exciting. Please try to get in touch with him. The goal is to see how we can support these investigations across Africa. So please make use of the opportunity. Just to give you a secret that those of them who are into the area of neuroscience are big boys. They have money. So just get in touch. He has some cash to spend. Dr. Kakari, I hope I'm not mistaken.
To an extent, not much. Unfortunately, we can't get those in our pocket, so we try to use the job.
Yeah, but the fact is that it needs to start from conversation. So please get in touch with him wherever he can support you. He has his network. He can connect you to those, perhaps those questions beyond his capability. So this platform is here to create such collaborations. So once again, thank you. We appreciate the opportunity and the time you have spent to give us this presentation, and Dr. Menoko for sterling moderation of this evening session. We appreciate the two of you. All right. Next month will be our next seminar series on the 27th of March. We do not have the specific topic, but we know we are going into agriculture. Our first seminar series was on computer science, information technology, and today we have had on medicine, and next will be on agriculture. The goal this year is to bring forward across all fields possibly to tell us what is happening in specific areas. So please join us again. And thank you, the audience, for staying with us this evening. All right, we'll call it a day here. Eman, do you want to do your normal job or you want to let us go? Eman likes taking pictures.
Yes. Fine.
All right, so please, you can show your faces, can show your camera. Emma wants to take picture of those who have attended this evening.
Yes, thanks.
Thank you very
Thank
much.
you.
Yeah, see you again next month. Bye.